Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. All right, welcome everybody to the Genetics Podcast. I'm very excited to be here today with Chris Hopkins, who's a serial biotech entrepreneur, and he's the CEO of Glafabra Therapeutics today, where they're developing a one-time cell-based therapy for enzyme deficiency disorders like Fabry, Pompe, Gaucher disease. I know that we're going to learn a lot about what the standard of care is today, but also what the future might look like for patients. So, Chris, thank you for joining us.
Well, thank you. Pleasure to be here.
Why don't we start with Glafabra? I'd love to just hear from your perspective what the problem is that got you so excited about the company and what you all are trying to do.
Well, sure. Fabry disease is a disease that causes patients to have heart failure and kidney failure because they're missing a key enzyme. And so without that enzyme, they get a buildup of toxic metabolites that causes this failure of these tissues. But another thing they have also a lot of CNS problems. The current standard of care has a difficulty in treating that. The current standard of care is enzyme replacement therapy. It does treat the cardiac and the kidney issues, the patients, but the challenge for the patient is they need to go in to the clinic and get the jab for the IV bag that is bringing in the enzyme replacement therapy. And they have to do that every other week. And that's really tedious for these patients. So when I heard about this particular therapy and I heard about this asset, I said, oh, wow, this thing it has dramatic effects. And there was a 5-year follow-up study that just got published last year that said 4 out of 5 patients were seeing a highly durable effect. But this therapy had been attempted to be commercialized once before by a company called AbraBio, but they ended up shelving the asset. And so when we saw this data, I saw this data coming out, I contacted the academic group in Canada that had published this result, and we got together and formed a company to try and get this asset essentially off the shelf and back into a clinical pipeline. So basically, we're taking and repositioning a shelved drug asset.
I mean, this immediately makes me think of one of our previous guests on the podcast, Reata Pharmaceuticals, that developed a Friedreich's ataxia program that they identified in an academic lab. And they created an amazing outcome for patients and obviously for their shareholders too. So maybe you could talk a little bit about how it works and what's the issue with the current standard of care, because it just from a patient's perspective, it sounds like going for I think you said weekly infusions, that sounds like a quality of life challenge right there. But are there durability issues? Does it not treat some of the core, underlying symptoms? Or you can talk a little bit about that.
Yeah. So basically, that you're hitting that about the current therapy. I mean, basically, thankfully, these patients have access to enzyme replacement therapy. So basically, enzyme is made in the lab and put into a bag, and then it's an infusion every other week. But that infusion is done generally in the clinic. So the patients need to build their lives around clinical visits, and that's quite tedious. So we have essentially an infusion, but instead of giving it every other week, it potentially is going to be given every 5 years. And the reason I say every 5 years and not a sort of a permanent cure is because for two sort of reasons. The first reason is that the patients in Canadian clinical trial, 4 out of 5, still going strong now, actually at 7 years.
Wow.
One of those 4 fizzled out at about 4 years out. Okay, so what that means is that there are, the durability of this is not maybe for some people will not be their lifetime. In fact, for probably, maybe the durability is only at around 5 to 6 years on average for most patients. But the beauty of a cell therapy approach, where what we do is we don't have the capsid antibody, capsids in the dose, so we don't develop anti-capsid antibodies. That's an issue with AAV, right?
Hmm.
It's a one and done, mainly because once you've done it, you can't redose it. This
And sorry, can you explain why? Yeah, why is that in this case that you don't have the antibodies and that you can redose?
Okay, so this cell therapy, a lot like CAR T manufacturing in the sense that what we do is we harvest cells from the patient, autologous. We then take those cells back to the lab and then modify them so that they now will express the missing enzyme. And then we put those cells back into the patient. So how do we modify them? We use a lentivector that has a transgene that encodes alpha-galactosidase. That lentivector was treated in the cells in the lab. We got that genomic integration, and then we wash away any
Mhm.
leftover capsids. So there's really basically no capsid around when we put those cells back into the patient. So there's really nothing, at least from a viral standpoint, for your immune system to react to. Now it might react a little bit, and in fact, we know it does react a little bit to these patients have seen enzyme replacement therapy, but there's a little bit of an immune reaction to the secreted enzyme from the cells. But that's a very short duration, and it stabilizes really quickly. So basically, there doesn't appear to be any immune reaction. I mean, it kind of makes sense because it's autologous. We're taking the patient's own
Yeah.
cells. They're not, it's not an allogenic therapy, it's an autologous therapy.
So this reminds me of Casgevy, the gene editing based sickle cell therapy. Maybe if because listeners will probably be familiar with that because it made front page news in late 2023, early 2024. Could you compare and contrast how that works and how we should think about what you're doing relative to that example?
Yeah, and I love that you brought that up because basically I just came back from a meeting on the Mesa, and I got a chance to sit down and chat with Victoria Gray, the very first patient ever dosed with
Right.
Casgevy. She's doing great. She's doing fantastic, and she probably has it for life, right? This solution, because she got a totally myeloablative procedure. Now, that's a risky procedure. Only some people can get it. It's basically you're eliminating that patient's bone marrow or hematopoietic stem cell reservoir is completely depleted, and you eliminate it and you replace it with essentially the patient's own cells, but now engineered to secrete the factor. And so that's you put that back into the patient as a fully bone marrow transplant replacement, right? In our therapy approach, that can last the patient's potentially her rest of her lifetime. She'll never need another treatment. But it's very harsh to get there, and only some patients can tolerate basically being put in the hospital for weeks. If you do a submyeloablative dose like we did in Canada, the team did that, and basically it was an outpatient procedure. You did not need to stay in the hospital after being given the therapy because your conditioning was very mild. It just basically what I like to call it is instead of a transplant, it's an augmentation. You're augmenting the existing reservoir. We did use melphalan in a submyeloablative dose to essentially make a little room in the hematopoietic niche. But it's not even, you may not even need to do that if you can tie this therapy into the mobilization. So basically, you need to mobilize the hematopoietic stem cells by giving patients GCF and Plexifor that will then lead to their hematopoietic stem cells getting into the bloodstream, and you harvest that by leukapheresis. But that right there has exposed the niche. If you turn around and get those cells processed and back into the patient very quickly, which is actually something we potentially could do within 24-hour turnaround time, maybe 48 hours, be able to get that back into the patients. The ability for those cells to now there's an empty reservoir, there's not an empty reservoir, there's some space for them
Yeah.
to come in a sort of submyeloablative way where they're going to now cohab right next to the rest of the unmodified hematopoietic stem cells. But if you've got good expression from your transgene, then the fraction of blood cells that are maybe 10% or 50% of the blood cells are coming from your transplant, they're still providing enough enzyme when they go to the various tissues in the body to secrete the enzyme, and the enzyme that's secreted then gets taken up by the neighboring cells in a process called cross correction, where they use that enzyme from essentially that donor cell and they now are picking up that enzyme and using it to control the buildup of toxic metabolites in that cell.
Yeah, this is amazing. I want to play it back just to make sure I'm getting it right, but also for everybody listening. So the we're basically pulling out hematopoietic stem cells using a lentiviral vector to edit those. And unlike I guess in the sickle cell case, you've got to replace everything or almost everything. Otherwise, you still have the sickling phenotype. But in this
Yes,
case,
right.
you need to get enough in there that you're restoring enough function. You don't have to have complete restoration because it's a loss of function, I guess, in the original case.
Exactly. And
Yeah,
that's the beauty of loss of function versus gain of function. So Casgevy is a CRISPR
yeah,
that's wiping out that sickling effect, and you kind of got to get rid of everyone. So
yes,
in a sense, you got to be fully myeloablative there. That's for dominant alleles. So recessive alleles, where especially if it's something that's sort of like a haploinsufficiency or something like that, where you just need to get a little more functionality in there. I mean, that's essentially kind of not on a cell and gene therapy space, but that's essentially what's going on with the treatment for cystic fibrosis. So the CFTR gene, they're just stabilizing a little bit more, so it reaches a threshold that the now no longer has it's now more homeostatic. So, if you just need enough enzyme to keep the toxic metabolites at bay.
yeah. Interesting. So you're starting with Fabry, but I suppose this could be applied to almost any case where, yeah, rather than me speculating, like how broad can this go? Like where is this anything that's on enzyme replacement therapy? Can we think about any haploinsufficient or recessive disease? How do you think about it?
Yeah, well, so there's 48 different lysosomal storage disorders. Lysosomal storage disorders are a great target because essentially the lysosome topologically is the same as the extracellular space, right? It's not the cytoplasmic. And there is an active transport mechanism to bring this in. And it's probably something from sort of an evolved anti-aging where the neighboring cell, or where two cells, right? One cell sort of loses the capacity to make a particular key metabolic enzyme, and the neighboring cell is able to either secrete it into the extracellular, potentially even through exosomes. So cytoplasmics could be treated potentially this way. But basically, the secreted enzyme, it's probably been an evolved system to have a little inefficiency of targeting the lysosome, and some of it gets thrown out into the extracellular space, where neighboring cells who have now from aging have lost the capacity can actually import that through this common mechanism of M6P, maltose 6 phosphate, decoration on the secreted enzyme. There's an active transport mechanism to bring that back into the lysosome. That's the
Mm.
cross correction phenomenon. 48 different genes are known that can potentially be rescued this way. So there are a lot of other targets, and this technology has a platform nature to it where really all we need to do is just swap out the transgene, what's called the transfer plasmid, and have a slightly different coding sequence, coding for a different enzyme, and everything else is identical. So it has the potential to be a platform technology with the FDA, and could have a lot more accelerated approval from that platform nature. So, I think Sarepta was one who's got a AAV platform designation with the FDA. So there's a couple different places to have that kind of capacity where basically all we're doing is just changing up a particular little segment of the coding sequence.
And I guess the other approach in the cell and gene therapy space here would be something like AAV, right? Maybe you can compare and contrast with an AAV gene therapy versus, but we're getting like a crash course in how many different
Sure.
ways there are to go after. I think I mentioned this to you before we started recording. I just did like a solo episode on AAV gene therapies, and I wish we'd done this in another order because I could have talked about this, but this is perfect. Maybe you can talk a little bit about how this compares.
Well, and it's actually one of my favorite comparisons. And basically, there's a couple different groups that have AAV therapy for Fabry disease out there. The challenge with AAV is it potentially could be one and done for a lifetime, maybe, but often it's not. And in case of AAV therapy for Fabry, there's some really good results coming out of Sangamo is the leading one there's a couple of that are developing AAV therapy. They were able to get a remat and now they're staring at a BLA, and they're able to go to market potentially very soon with an AAV therapy for Fabry. Now it works amazingly well, right? It looks like it's got good control of some of the toxic metabolites, looking at the biomarkers there. But it has the chance of fizzling out. From a different therapy, I had a patient, different, not Sangamo, one of the others. One of the patients came to me and said, and 2 years out after being on the clinical trial, they said, "Hey, I'm not able to take my AV again because now I have a huge antibody reaction to the AV. Somebody told me about you. Can I get in on your trials?" So with our therapy, because we could certainly take them on, and the 4 out of 5 patients where one patient fizzled out after 4 years, that patient I interviewed, they absolutely want to get back in on the clinical trial, and we could redose them.
Hmm.
So we're able to redose, and that's one of the advantages. So AV, like us, has really long duration, years long duration from one dose, and has large effect that can have a lot of effect in tissues. But that's actually one of the challenges with both ERT and AAVs getting across the blood-brain barrier. And in an inflamed brain, the white blood cells can get in there, especially T cells can get in there and be able to secrete the missing enzyme. And in fact, anecdotally, we believe that in interviewing the patients where they say, "Oh, my brain fog's gone, I feel wonderful." We want to be measuring those clinical endpoints, those CNS endpoints in the clinical trial. So our goal is to start a clinical trial. We're raising money to try and get this therapy into an IND, get an open IND, hopefully be able to start treating patients, a year to 2 years from now. The CMC hurdle is huge here to try and get across, but we've already made this, so we're trying to try and leverage the Canadian CTA and to get to the IND, and then be able to bring this to patients and hopefully start a clinical trial in the US next year to maybe 18 months from now. Maybe it all depends on whether we get good funding. Funding will go fast.
Yeah, so let's assume that the person or people who want to fund this are listening and write you that check that you need. What does it look like from here? So you have the 5 patients that have taken part in the academic study. So what is, roughly speaking, the path to getting this through clinical development and into approval look like? Obviously, with all the caveats that we don't know, and it depends what the FDA says and all those questions.
Right.
But what could it look like?
And I wish I had my pitch deck. I could throw it up to you right now, but we've got essentially a pro forma forecast. What do we need, when, and where? So we're asking for $500,000 pre-seed right now. That's going to do 2 main things. It's going to help us get our orphan drug designation, which gets us sort of a lock-in on those accelerated approval pathways and a 7-year market exclusivity. And then we're also going to use that to get an interact meeting with the FDA, and that's really where we hope to get a lot of clarity. So we're building our TPP right now, to target product profile. We're doing a lot of things to sort of get the data that we want to bring to the FDA and say, "Hey, look, there's the Canadian CTA. Look how good this was. What do we need to do to get this into trials in the US? What kind of updates?" It was done 10 years ago. It was one of the very first types of therapies that was done for cell therapy for a rare disease. And so, there's lots of places you could update. There might be great improvements in the transfer plasmid that might give you much higher packing efficiency. But do we want to go with the original one? So we're trying to figure out the trade-off. But basically, how do we get this trial repeated in the US as fast as possible so we can get patients treated and get the same results that have been seen in these other studies, and be able to bring to market a therapy for these patients. So then there's the CMC side to get the open IND. That's going to cost us about $4 million in direct costs, and then there's other costs. So we think that it's about $500,000 to get positioned and de-risked with the having had a meeting with the FDA. Then beyond that, we're going to need about $10 million to get to phase 2 efficacy data. And if we're going to bring our other programs that are in Pompeii, if we're going to bring those 2 forward as well, then we're going to need a total of about $50 million to get all of those programs into the clinic, demonstrating efficacy, demonstrating the platform nature, and then to develop that nice acquisition capacity with pharma. I will say that amazing. 24 companies have exited in cell and gene therapy to pharma, and the average exit price for those ones that have been bought was $1.3 billion. So pretty good exits if you can get there.
Yes, is this, because sometimes gene therapy trials costs like $100 million for just 10 patients or something because the manufacturing is so incredibly expensive. Is that not as much of an issue here?
Well, it is a bit of an issue, but it's not as expensive as 100 million. I mean, it's more like 10 million per indication
Right.
to be able to, if you're doing an orphan indication, because then your population size is low. I mean, if you have to dose, if you some of the cancers are going to not be rare, and then they won't get the advantage of an orphan drug designation. Accelerated approvals, lower patients' population size, and adaptability in the studies. Like if you have to do 1,000 patients that have it and 1,000 healthy patients, you have to recruit them. I mean, my gosh, that costs a lot of money. Here we're talking about a study where well, we're working in partnership with Uncommon Cures. And they've told us that they think that we might be able to get away with as low as 15 patients. Now, the predicates are out there with Sangamo and a few of the others that they're using 50 patients, and they've all got orphan drug designation. So what we think is that we should be able to do this with a sufficient. I mean, we're already powered really well, surprisingly well in those 5 patients.
Yeah.
So turn that into 50, and now we're 10xing it. Now we're really going to get powered. So we should be able to see really significant results and hope maybe enough power to be essentially called the pivotal trials, right? So that happens, right? You get the remat. And so anyway, there's a lot of angles to this, but the accelerated approvals with an orphan drug keep that cost down.
Yeah. And what do you need to compare against here? Is it against enzyme replacement therapy, or are there patients like in parts of the world, for example, that don't have
Uh,
access to any therapy, and you have the with rare diseases you can often pursue natural history control arms. I'm just curious to hear how you think about like what you compare against, because it's not always an easy question, right?
Yeah, many of these patients that would be going in these trials, like they were in the Canadian trial, have been on enzyme replacement therapy. We took them off. They saw a 41% jump in the level of
Mm.
toxic metabolites after about a month to 2 months being off. We gave them the cell therapy in Canada, dropped to a 48% reduction in the level of toxic metabolites, which was lower than the starting place. And then then we went to those patients and measured them 5 years later, and after they were still in control of their toxic metabolites and at a dose that was lower than the ERT 5 years later after that first initial dose. So it has this really long durable effect. Now the size of that population, those five, and then the other studies. Well, what is the current enzyme replacement market size? Is kind of what you're getting to. There, if you look at the Sanofi's registry on clinicaltrials.gov, it indicates there's about 10,000 people in there. Probably most of those are getting enzyme replacement therapy. So there's, 10,000 people are in there getting treated. It's expensive right now for them. Average patient somewhere between, depending on the size of the person, anywhere from 500,000 to 1 million a year.
Wow.
So, it's a pretty expensive procedure. So there are a lot of people can't get it. If 10,000 people are sort of getting it right now, and we're thinking that number's the current treatment with all the other suppliers out there, and Takeda sort of has the European with their therapy, but the Fabrazyme's mostly in the US. So we'll be trying to do a whole bunch of market analysis, like where's the market. But what's interesting, go into Nomad and ask how many of these pathogenic alleles. Most of these pathogenic alleles are pretty Mendelian. They're pretty much going to you're going to get Fabry if you happen to have that. Those pathogenic alleles are like 200 of them in Nomad, which is a healthy population database of 800,000. So that means there's one in 4,000, potentially 1 in 4,000 people are doomed to suffer from Fabry disease around the world. That's a big number. We're talking like a million people. And so only 10,000 has been treated right now, or what's the diagnosed pool? Much lower, like an order of magnitude lower. So there's a lot of undiagnosed people out there. And then the diagnosed pool, there's a fraction of that that's been being treated.
Wow. I mean, it's fascinating to understand all the different options as well that patients are going to have in 5, 10 years time. And it's really encouraging, honestly, because we don't want to just have only one option, right? We want to be able to redose, try something different. Sometimes it's very puzzling why something doesn't work or stops working for a patient, right? So understanding systematically why and then feeding that back into the pipeline. I wasn't really aware of this underdiagnosis problem as well. And so that's another
Well,
layer
that's
to the
across
issue.
all rare diseases. This is why when people are running away from rare disease, what are you thinking? Look at what GeneDx is doing in the baby-seq space. They're coming up with numbers on the baby-seq that haven't been essentially filtered, and the numbers instead of 1 in 4,000 are coming out to 1 in 2,000. So it may even be
Mm.
higher than that. So things that were and the here's another thing about sort of the frequency before it starts getting filtered out by selection pressures. Just about any rare disease gene is going to be, it won't get sort of selected against until it's 1 in 100,000, right? If it's, depending on essentiality and a lot of things. I mean, it's pretty variable, but when people say, oh, this gene here we've only got 4 patients in the world that have it, it's ultra rare. Well, guess what? It's just underdiagnosed because it won't get selected for in the population. The natural mutation frequency will sort of drive it to be at about 1 in 100,000. So there's any given rare disease probably has about 1,000 to 10,000 hiding out there. They just haven't been diagnosed.
Hmm. What do you think about where we are as an industry more generally? There's funding challenges, but also the science is probably more exciting than ever. Are there things that if we look outside of what you're doing, that you think there's a lot of promise there and we should be doubling down in that area? And also if there are things that are there, that it's genuinely overhyped now. We need to move away from it. I'm interested to get your perspective because this isn't your first company by any stretch. You've been working in this industry for, I think, 20, 25 years at this point. So I'd love to get your perspective on it.
Yeah, well, LNPs and RNA, those are really, really intriguing. I mean, and we taught that we've experienced it with COVID
Yeah.
vaccines. Those things can really make a difference. Literally tens of millions of people were pretty much saved from a death sentence there. So these things can be very impactful. They're still very challenging to kind of deliver, but and there's some immunogenicity problems with some of the LNPs, but they're really exciting. Another area that's really exciting is allo. So we're autologous, patient's own derived, but if allo can get to the point where the sources can be as low immunogenicity as possible, the graft versus host problem will not be so bad. And then we'll really see something that both is economic, because it's expensive to take a patient's own cells and to,
Yeah.
multiply them and everything. If you could make an allo that could do the same thing as an autologous and now be able to grow it up to an infinite supply, you'll have a much more cost-effective approach to bring patients. And if you can, it's capitalism. It's cost of goods sold. If it costs me 100,000 to make it, and I want to charge 200,000, that's nice capitalism, right? But if it costs me 10,000, I might be able to charge, maybe 30,000, make a little bit better
Everybody's
margin.
happy. Yeah.
And everybody's and we're treating more patients. Right now, that's the big problem, is only a very select few, maybe it's only like 1% or 0.1% are going to have access to the current pricing. And so if we can, think about that, right? So if you could, drop the costs by, tenfold and treat 100 times more patients, that's a bigger haul from the market. I mean, that from a capitalistic standpoint,
Yeah.
let's find those cost of goods sold efficiencies so we can get the larger market grabs. What's that pricing optimum? So there should be the reductions in pricing and ways to make these more efficient. I think that's coming. That's what's exciting. That's a lot of effort that needs to, be done, is to sort of automate. And, with AI coming on, there's going to be a lot of efficiencies grabbed from automation to reduce the cost of goods sold, and to be able to not make it such an elitist medicine and generalized access for everybody. I think that's coming. I mean, that just happens. Look at the antibody market, right? It took about 10 years before it started taking off, but now it's really taken off, right? Cell and gene therapies is kind of, probably going to follow that same footprint. It's going to find these efficiencies, find these niches and these multiple applications. Even something like Fabry, it's potentially something that happens. I told you about the aging concept, right? And loss of metabolic enzymes. Maybe these therapies on a this is a pretty wacky, right, kind of idea, but how many ways can you take what's done on rare and then apply it to the general populace and have benefit? There might be an anti-aging benefit of something like this, right? These kind of therapies.
Yeah, I can totally buy it. I think the more we're able to like fine tune this on a cellular level, the more that becomes possible and not so much science fiction, right? The idea of an obesity magic pill 20 years ago was pretty magical, but we have it today, and it's not perfect, but it's amazing what science can do if people bang their heads against it for a little while.
There's some people going after metabolic therapies in the similar way, where we install essentially cells in you that will
Yes.
essentially drive you to have better metabolic control.
Yeah, I heard a very interesting idea recently. I'm trying to remember where I heard it because it's not my idea, but I'm just going to try to reproduce it faithfully here. Of you've got in the immunoprivileged parts of the body, the eyes, the central nervous system, those are probably going to be a great place for AAV and other kinds of viral gene therapies because you don't have as much of an immune system issue there. But in other parts of the body, you almost could think about the reverse strategy, which is you utilize the fact that the immune system is everywhere, and if you can design a genetically engineered immune cell that sends the right cargo to the right place in the body to fix whatever is going on, then you almost have the immune active part of the body where you want to use approaches like you are, where you're harnessing the fact that you're taking the patient's own cells, you're reprogramming them in some way, and you're putting them back in, and you're not getting the immune system excited and in a bad way. But then in the immunoprivileged or the immunoprivileged parts of the body, you actually can do the reverse and have a lot more degrees of freedom. So I thought it was an interesting way to think about it. We don't have the technology yet to do all of that, but it made sense to me, just from a first principles perspective.
Well, for sure. I mean, it's exciting what's coming on. Meeting on the Mesa that I just came from, I just had a lot of really interesting, innovative approaches, and it just seems to be exploding right now. Sadly, the funding, at least in the U.S., is under threat. Hopefully, that's the seed engine, the NIH, and hopefully we can, lobby our congressmen to support that I guess if they'll ever reconvene, but
Yeah. Yeah, that's right. It is. No, I think the longer it goes on, the more the uncertainty becomes problematic, right?
yeah, yeah.
I want to finish up on a very interesting lighter note. I understand that you invented a technique to milk venomous snails in one of your, I assume this was not done at Glafabra Therapeutics, but in a previous occupation. So tell me more about that because I'm very interested.
All right. Well, that's all right. I love that story, mainly because it's just an undergrad way back, we're talking about 30 years ago. I was working in a lab of Toto Oliveira, and he was studying venomous cone snails, a snail that eats fish by stinging them with a venom. If you haven't seen
I
it,
love that.
go on YouTube, check out the videos. They're pretty crazy. So basically, what it is this little marine snail that's in the tropical reefs. It slithers around in the sand, and it sends out a little proboscis that is essentially a tongue, really long tongue that looks like a worm. And a fish comes down and says, "Oh, tasty worm." But what's inside that is a harpoon, and they have some of the most potent neurotoxins that as soon as they touch that fish, they fire that harpoon and they squirt a bolus of these neurotoxins into the fish, and it paralyzes them. Well, how did we harvest those toxins? We killed the animals and dissected out that tissue that has the toxins, and then we isolated the toxins. Pretty destructive, right? And these are kind of maybe even endangered species.
Yeah.
So the question well, you can milk a snake, you can milk a spider. Can you milk a snail? So I started thinking about it. Okay, well, I told you about how they sting through the fish and squirt the venom in. I started thinking about it. I was like, okay, what can we do? What kind of membrane is out there that is really good at not getting torn and will sort of allow us to capture that bolus of venom? And so I got a purchase order from the University of Utah. I went down to the local supermarket, went over to the drug section, found a box that didn't have any lubricant. Brought it back to the
That is exactly where my brain was going. Yeah.
brought it back to the cashier and said, "Here's a purchase order." And she's like, "What's a purchase order?" She calls over her manager and so on. So we have this pretty interesting dialogue of me, getting my purchase done. So I head back to the lab and I blow up one of those little condoms, And I've got all you put some fish nearby, and the cone snails will get excited and they'll start sending the proboscis out, trying to get the fish to sting. And I'm sitting there, and I've got the condom on a forceps, and I'm bopping on the end of the proboscis, and they're not stinging it. And I'm like, God, what do we need to do? And then I had a sort of a eureka moment. They probably have chemoreceptors on the end of their proboscis where they've got to taste the fish. So I took a little fillet skin of fish, duct taped it to my condom, and instantly they just fired it through.
Oh my gosh.
And it's a barbed harpoon. And so I let go of everything, and the condom shoots to the top of the tank, the marine tank, and the snails sitting there dangling underneath. And you could see it in the condom was the big
venom.
Of venom. Well, we then figured out how to turn that into a little Eppendorf thing, and actually it turned out latex gloves were just fine. And we figured out how to do that to create and harvest a ton of venom. So that's
amazing. What do you want to do? So I imagine you want to just understand the venom as a scientist, like what's going on here. But what else, if anything?
Well, one of those venoms that was isolated was a cardiological venom that actually ended up, they're used as tools in research, but one of them was being explored for an anti-arrhythmia drug.
Fascinating.
It's injectables, so in that sense, it's not as good as a pill, but it's still, that one I don't think really took off, mainly because it was, injectables are just a little more, if you have to keep re-injecting, that's frustrating for patients. As in the case of Fabry, where you have to continue to keep getting hooked up. Those are patients don't like to be jabbed multiple times. And so that kind of took away that, but it was a very dramatic effect, I guess, on an anti-arrhythmia neurotox or drug that was essentially preventing that it was coming out of the venoms. And then just really good research tools for understanding the different types of receptors and so on. That was the utility of where Toto Oliveira went with his lab.
That's an amazing story. I love that. I had to ask about it because I saw it on on my background notes. As we wrap up here, is there anything that you wanted to cover that we didn't, or if not, is there something that people listening can help you with, or that you want to leave them with as a message?
Yeah, so I guess for if the audience is sort of like wanting to be inspired about taking the leap from academia to industry, it has a scariness, but it's amazing. And what you want to do is just surround yourself with those teams. There's some really good incubators out there, and get into those and take your concept and make it happen. Don't be afraid, go for it. For me, I have a little bit of, de-risking. Was my dad had done that a long time ago. Now we're talking about 40 years ago. He had started his own company when I was just a kid. So I had that example, but that also, you get those examples, you see those examples in some of your advisors. So if you got something you want to think's got commercial potential, don't be afraid to jump on and chase it. It's a fun ride. It's a crazy ride. There's a lot more learning. I went back and got my MBA because that was really critical to getting some success. But it's still an awesome ride.
Yeah, I couldn't agree more. Well, thank you. I really appreciate you taking the time and also all the amazing work you all are doing. So I look forward to catching up with you again soon.
Thank you. Appreciate it.
Thanks, everyone, as always, and we'll see you next time. Thanks, as always, for tuning in to the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sanogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sanogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show. Jane Reddy Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.